Well, welcome back. We are in Chapter 13, the Microbe-Human Interactions. This is from the 6th edition of Microbiology: A Systems Approach by Marjorie Kelly Cowan and Heidi Smith. And we're going to be talking about the microbe-human interactions today. So for the last couple of chapters, last unit or so, we've been looking at the microbe-host interactions and the host microbe interactions and the microbe drug interactions and the drug microbe interactions and the drug human interactions. So this time it's the microbe. What does the microbe do to the host? So this is looking at the disease process, and there are basically 3 sections to this chapter. Section 1 is looking at the normal microbiota, and it's kind of an orientation and introduction with some terminology about disease. And so we're looking at the microbiota here, the microbiome, and what we know about the microbiome right now. And so this is a kind of a changing area. This has been changing over the last 20 years. A lot of research has gone into the human microbiome, and a lot more has been learned. And so this is going to have some updated information. And then section 2 is talking about the progress of disease, so pathogenesis. And if you've had pathophysiology or some other nursing classes, this may sound very familiar, but we're going to look at this again from the microbe. What does the microbe do to the host? So specifically looking at what the microbe does to the host. And in the 3rd section, we will be looking at the epidemiology. So a lot of terminology. In fact, in my study guide, I have up to EE in terms of terms. And so without further ado, we'll be looking at Chapter 13. So as I previously said, these are the 3 sections we're going to be looking at for this chapter. The first section is looking at the overall terminology having to do with the host microbe-host interactions, and then pathogenesis, and then epidemiology. So again, like in Chapter 12, we looked at what the drugs, the chemotherapeutics, did to the host and to the microbe. So drug-host and drug-microbe interactions, and then we looked at what the microbe did to resist the drug. And in Chapter 14 and 15, we looked at how the host responded to the microbe. And so in Chapter 13, we're looking at what the microbe does to the host. So what is this progress of infection or pathogenesis? So that's kind of the way I look at these 3 chapters, and they're all, trying to figure out how this then ends up in infection, I think is a really big thing here in Chapter 13. So the learning outcomes are to differentiate among the terms colonization, infection, and disease. So those are not all the same thing. And then second, to identify the sites where normal microbiota are found, and discuss how the Human Microbiome Project has changed our understanding of the normal biota. So colonization, infection, and disease. So infection is when it's a condition in which pathogenic microorganisms penetrate the host defenses. And so they have penetrated the host defenses, they've entered the tissues, and they are multiplying. So they're present. They're just, that infection means you have pathogens present. It doesn't mean anything about what they're doing to the host, they're just present. A pathogenic state or pathologic state is when you have a disruption of tissues and organs, and that would be kind of like the term that I would use for disease. So it results in disease. So you have the cumulative effects of infection damage. So disease, any deviation from health, and it would include these factors that can be contributing to the disease. You can have infections previously, you could have genetic resistance or not, and aging can result in different effects, and malfunctions of organs or systems. And the infectious disease with a disruption of the tissues or organs, that's what, again, I would consider the definition of disease caused by microbes or their products. And so those are, again, some different definitions of that infectious disease. And then the resident biota or normal biota or normal microbiota or normal flora, they have a lot of different names for this. And I have always called it normal flora because the bacteria used to be part of the plant kingdom. And so if you have flora, that's plants, and if you have fauna, that's animals. And so the resident biota or microbiota is what I typically call it. Those are microorganisms that live in and on you. So they're living on the body, on the skin, and they're in the body in various organs, and places that we previously thought were sterile are actually not sterile. And in actuality, the normal microbiota makes up about 3 times more cells than human cells in your body. And so they have a profound effect. So it doesn't just include bacteria, although we're going to focus on that mostly, bacteria. Fungi, protozoans, viruses, all of these things are part of the resident biota. Even small like mites and other kinds of animals that are insects, arachnids, arthropods of some sort that may be living on your body. And I know that makes you itch just to think about it, but they're part of this microbiota as well. So it's something we can't really see. The relationships among these resident, transient, and disease-causing microbes and the human host. This is a really great infographic. And I'm going to hopefully zoom in on this a little bit and we'll talk about. So if you see it in green, that means it's a resident microbiota and it's friendly. Pathogens or potential pathogens in red, and then the transient flora or microbes. See, I keep going back and forth between flora and biota. But the transient microbes could be present as well. So all 3 of these things can be present, and we call this the holobiont. So look at this term here in particular, the holobiont. That's you. You are a colony of organisms. You're colonized by all these different microorganisms, and your cells are part of that colony. Or maybe it's better to say that these microbes are part of your body in some way. But the potential pathogens and the transients that are so red and purple here are they're going to be gone. So you wash your hands and they can simply exit the body and then they're gone. Potential pathogens and sometimes the resident microbiota, so some of the green, can be present in places that are inside your body and can move away from areas that they're supposed to be in if they are a microbiota. And some of those infections will be asymptomatic, so there's no symptoms, but they're present. Remember, the infection means they're present in or on your body. So they could be gone, they could be washed away, and your immune system could be clearing them. So that would be one possibility. Or the infection could expand and proliferate and spread through the body, and the microbes can cause disease. So you could have some symptoms, and if your immune system can't handle it, and for whatever reason this microbe is super pathogenic, occasionally that leads to death. And this could be something like pneumonia or sepsis, or it could be COVID. I mean, there's a lot of different reasons for that, but so when it causes damage, that's when you have disease. So if the infection spreads and it causes damage, then you have disease. And if it continues and it's not cleared, then it can cause death. But usually they are eliminated and they're gone. They're out of there. Some of these may eventually become part, so these ones that are disease-causing could be part of the holobiont. They may be kept at like lower levels, so you can see the few of these potential pathogens throughout the body that can be part of the microbiota, you know, like Clostridium or some other different staph bacteria. But as long as they're kept and they're under control by the rest of the microbiota and prevented from growing too terribly much, they're not going to cause problems. So that's kind of the fact that there are so many more green here. The resident microbes, they prevent the red ones, the potential pathogens, from really getting out of control. So that is kind of the way to look at how all these things are related. So there's a lot of reasons why something could go from, just being asymptomatic to becoming disease-causing, or why it might just be eliminated, and we'll talk about that in a little bit. So the Human Microbiome Project, this was started in the early 2000s, and human cells, and the preliminary results were that the human cells contain around 21,000 protein encoding genes. That microbes that inhabit the humans contain around 8 million genes. So there's a lot of genes out there, and a lot more of them are not human genes. And those have been identified through various methods of biotechnology where they've been able to sequence the microbe, or sequence the human samples from humans and different places in the body. So we have a lot of microbes in places we used to think were sterile. That's a big part of this that is new. And I've had to change my questions in my quizzes, because places that I had previously said were sterile, we're finding out actually have potential pathogens in them. And all healthy people contain some potentially dangerous pathogens, but they're in such low numbers because your human microbiota, your normal microbiota, is keeping them in check. So that's some of the big things about the microbiome. So the importance of gut microbiota, we have found there's a lot of differences in people who have Crohn's disease, obesity, heart disease, autism, asthma, diabetes, mood. And so the consistency, the type of microbes that are in the gut biota in people with these different diseases is significantly different. So we know that. Whether that is a cause or not, all we can say really is that they're associated with the differences in the disease and risk factors for those diseases. So current understanding of the sites containing microbiota, we have always kind of known that these particular locations have more normal microbiota. So the skin, mucous membranes, that's a no-brainer. It's on the outside, and mucous membranes are exposed to the outside. Upper respiratory tract is exposed to the outside because that's where you breathe things in. Gastrointestinal tract, especially the mouth, outer portion of the urethra, external genitalia, vaginal tract, the external ear and external eye, like the conjunctiva and the eyelids. So the one thing that they all have in common, yes. If you were thinking about that question, the answer would be the one thing they have in common is that they all have some connection to the outside, and there's an orifice allowing the microbes to get into this internal spaces, but this is like the portal. And so additional sites that we said, no way, there are not microbes there because we've got like protections against it, but we are finding the lower respiratory tract, the lungs, the bladder and urine and breast milk and amniotic fluid in the fetus. All of these have at least some microbiota, or at least they have detected their DNA. And then sites that microbiota has been detected, or in which DNA from microbiota has been detected, would be the brain and the bloodstream. And again, all of these sites that are listed on this page, previously we understood that they were sterile. We used to think that the breast milk was sterile until it got outside of the body. Bladder and urine were sterile until the urine left the body. Amniotic fluid, sterile until it leaves the body. Blood, sterile until it leaves the body. And so now we're finding not so much. So those are actually places that contain, at least they have identified the DNA from that area, or even sometimes microbiota. So how do we obtain the microbiota? And there are so many really good videos for this, and so I'm going to highlight a video and then just point you to it in the playlist. But this is, there's one about how babies get their microbiota, and is there a difference between C-section and vaginal? And there definitely is in the microbiota, that babies who are born by C-section are very different than the babies who are born, their microbiota is different than babies who are born vaginally. So I highly suggest that you watch that video following this video and then come back and watch it. But the benefits of the microbiota is that it does influence the development of organs. And so without those microbes, the organs don't develop properly. They help to prevent the outgrowth of harmful microorganisms. And so there's this microbial antagonism. So there's so many good microbes that the bad microbes don't have a foothold, and they can't establish themselves in the body. And microbes are in a steady, established relationship are unlikely to be displaced by incoming microbes. So the incoming microbes aren't, they're already established. Like I'm squatters' rights, and they've been here for longer, and there's more of them, and so new ones coming in, unless they're really big bad ones, they don't displace the incoming microbe. They are not displaced by incoming microbes.